When it comes to competent cells, there is so much to know, so many types and different qualities for different purposes. But in this video, we're going to look very specifically at E. coli cloning strains and learn about what they are and what makes them good for cloning in the first place. So let's begin with what cloning strains are. These are host cells used for cloning to copy specific DNA sequences. They're chosen for their ability to incorporate DNA, replicate it, and maintain its stability. In order for cloning strains to work well, certain qualities really help. These include their ability to grow quickly in solid media, high transformation efficiency, the ability to maintain and replicate the vector and clone transgene without incurring mutations, having visual ways to distinguish from desired from undesired clones, sensitivity to most antibiotics, so selectable markers can be used for screening, and the absence of endonucleases or enzymes that would break down your vector or insert. E. coli on its own naturally has a lot of qualities that make it good for cloning. It grows fast with a doubling time of only 20 minutes. It's already sensitive to most antibiotics, so antibiotic resistance would then require the presence of certain genes that can be part of the vector to be used as a selectable marker. But then there have been modified strains that give it the other characteristics such as lacking endonucleases and enzymes for homologous recombination. These traits in cloning strains prevent unwanted mutations in the cloned DNA. Two examples of E. coli cloning strains are GoldBio's GB5-alpha and GB10B cells. But for the rest of the video, though, I'll mostly just refer to them as 5-alpha cells or 10B cells. Each of these cells have some qualities in common and other qualities unique to the strain that make it ideal cloning strains for certain situations. For 5-alpha, there are mutations in the REC-A1 gene and END-A1 gene that ensures the plasmid you're introducing doesn't recombine with the host genome and it's not degraded by endonucleases. It's also engineered for alpha complementation used in blue-white screening. This is important for visually distinguishing your desired versus undesired clones. We actually have some other videos on blue-white screening and alpha complementation, so we'll link a playlist in the description below. Let's take a look, though, at some of those mutations and qualities. The first one we mentioned was the REC-A1 mutation. REC-A facilitates homologous recombination, but when you're cloning, you don't want that recombination between your plasmid construct and the host genome. If a strain expresses REC-A, homologous recombination might occur between your plasmid you introduced and your cell's genome. What you really want is for the vector sequence to remain intact exactly as it was when you introduced it. So the REC-A1 mutation ensures this won't happen. The other important mutation is the END-A1 mutation. Endonuclease 1 cleaves double-stranded DNA. With your vector being double-stranded, this means it would be cut by endonuclease 1 in your host cell. 5-alpha cells don't have endonuclease 1 due to the END-A1 mutation, and that means your vector and your gene insert won't be degraded within the cell. Now that we've talked about the properties of 5-alpha cells, we can look at the qualities of 10B cells that make it great for cloning. For starters, 10B cells also have REC-A1 and END-A1 mutations, so you won't have to worry about recombination or endonucleases degrading your insert. It is also engineered for alpha complementation and blue-white screening. But 10B has some other qualities that make it suited for other cloning situations. First, this strain lacks enzymes that normally cleave methylated DNA, allowing you to clone eukaryotic DNA, which are often methylated. 10B cells also have a characteristic that helps with cloning larger vectors. So let's look at each of these characteristics a little more. The first is the quality that makes it suitable for eukaryotic DNA, and this comes from the MDRS gene loci deletion in 10B cells. The MDRS genes encode enzymes that cleave methylated DNA, which is common in eukaryotic DNA. With the loci removed, your methylated DNA is not at risk anymore. The other important quality of 10B cells is the ability to handle larger plasmids or another vector like a bacterial artificial chromosome, or BAC. The accepted reason behind this is that 10B cells have constitutive deoxyribose synthesis. Right now, there isn't a lot of information between the correlation of deoxyribose synthesis and higher efficiency with larger vectors, We just know that it leads to a more seamless replication of larger vectors. While these two strains have a lot of properties for cloning, there are some limitations. One is that they have methylase enzymes that methylate adenine and cytosine by a site-specific manner. So if you introduce a plasmid with or without an insert gene fragment, if you're using these strains, the DNA might get methylated. Thankfully, there are many different cloning strains out there, and to overcome this limitation, JM110 is an example of an alternative cloning strain, this one lacking those methylases. But in the end, 5-alpha and 10B cells are very common and very useful cloning strains within biotechnology. Both are broadly suitable for a wide range of cloning applications and should be considered for many of your cloning needs. If you've been researching competent cells, whether it's which ones to use or what's affordable, Gold Bio has a lot of resources on our website, along with a really amazing selection of cells for you to take a look at. We'll even have some links in the description to help point you in the right direction. I hope that this video was helpful, and thank you so much for watching.